It is Spinal Anatomy. It is week 5. It's Wednesday, winter 2021. Off we go. Nucleus Pulposus. We know a little bit about this, but let's get into this thing a little bit more. Histology of the Nucleus Pulposus. I mentioned this before, but here it is officially. So humans once you get over about 10 years of age, the cells of the Nucleus Pulposus change from their original notochordal cells into more chondrocytes, fibroblast type cells. Kind of a weird mixture of chondrocytes, fibroblasts. They do the same thing. They all make glycosaminoglycans, ground substance, type 2 collagen. But the thing is, the body's immune system might not know who these new cells are that pop up here. When you're under the age of 10 years old, remember we said last time that notochord cells are the ones that make the nucleus pulposus. And so cells need something to sit in, so that's called a ground substance by histologists. Physiologists tend to call it an interstitium because that's what capillaries dump their oxygen and glucose and nitrogen load into this ground substance or interstitium, and then these nutrients have to diffuse over to feed the cells. It's also produced by Nucleus Pulposus cells, just like glycosaminoglycans. And it's not a water, it's a kind of like a toothpaste consistency, but it's more of a gel. Not as stiff as Jello, but it's semi-water, but it's more like a kind of a wet toothpaste. And that's what everything sits in. Cells and the collagen fibers, everything sits in this ground substance. And technically it is the interstitium, as I said. It's a slippery toothpaste. In the Nucleus Pulposus, which we're talking about, it's because of the glycosaminoglycans, they grab water. Glycosaminoglycans, which we will get into the weeds on them, they're very hydrophilic and they grab water. And therefore, because the population in the nucleus is so high, the nucleus is about 80% water. Right, Annulus Fibrosus, we're pretty good at this, right? It corrals the Nucleus Pulposus. It's made of, the official number is 15 sheets. Some authors say 20, but we'll go with Bogduk here. 15 sheets of collagen make up the lamellae. Now, here's something you may not know. The lamellae even pass through the cartilaginous vertebral endplate to completely encircle the nucleus. So let's look at this cartoon, and this is exaggerated. Let me see if my drawing tools are going to work here. And so you can see the lamellae here. See how they completely encircle the nucleus pulposus? This region right here, this is the cartilaginous vertebral endplate. All of this region above it, that's the bony vertebral endplate. Even bony out on the ends here, the ring apophysis is, it's supported by the bony endplate. So, and that's where, did we talk about marrow cavities? I can't remember if that was last time or it's coming, but there's marrow cavities here. And remember the microcirculation, the capillaries dump their load off, their oxygen and their glucose, and they diffuse at nighttime in to feed the nucleus and to feed the inner annulus as well. But notice how the lamellae completely corral this nucleus pulposus. Okay, in an overhead view or an axial view, are lamellae complete circles? So in other words, if this is overhead view, here's the nucleus pulposus. Here's the annulus fibrosus. Here's the thecal sac and the exiting nerve roots here. Those are dorsal root ganglia. So do these lamellae, let's see, draw them in orange. Do these lamellae do this? Do they completely circle the nucleus? And are they complete circles? Well, it turns out that's not really true. almost 50% of them, 40%, are incomplete. And so maybe those first two I drew, they work. But maybe the next one is incomplete, so it leaves a hole, right? So it may look like that. Okay, and then the next one, if you have bad luck, maybe the next one is like that as well. Anybody see a problem coming? Yeah, and this incompleteness tends to happen in the worst possible place because we have the nerve roots right here. We'll draw this. We'll say this is the L5 disc, and there's the S1. Looks like Mickey Mouse. There's the S1 nerve roots I just drew. Here's the L5 nerve roots. That's the L5 disc. But you see what happens. So if you fall down on your butt and get an axial load injury, you can start the pressure increases, and let's make this pink. So the pressure increases enough, you can rip through these first two lamellae, and then it's easy sailing from there on out in our little model. And so it's very easy for you to develop an annular tear of the disc because of this, committee problem. Why the committee didn't make these complete, or at least make them incomplete in the front, I don't know. But it does, it's another reason why these posterolateral herniations occur. And of course, that nuclear material can come right out and it can crush the traversing and the exiting nerve root. And then these patients typically have severe sciatica. And maybe it doesn't come out. Maybe you're just left with an annular tear. That in and of itself can be devastating. All right, so committee error. All right, and there's everything, kind of Bogduk's picture of it, how they're incomplete, and boy, this one's really incomplete, even worse than I drew it. So that's why the human has so many posterolateral disc herniations. And there's the herniation. It's ripped right through this area, and big trouble, right? Because we know that in the outer portion, that's where the sinuvertebral nerves live, right? And so in some but not all people, this nucleus pulposus, when it gets out here, it causes a horrible inflammation, and these nerve roots get swollen, and they send off pain signals up to the brain saying, oh my God, my back is killing me, my leg is killing me, what's going on? So here's an old patient of mine, 2017. Can't even actually totally remember the case, but remember these sides are reversed, so this would be left over here when you're looking at images. We're really looking from the bottom up. There would be a cut line cutting right through this disc over here, and whatever this cut line goes through is what you see over here. So we see a cut through the disc, and we see a huge disc herniation here, completely smashing the thecal sac and the traversing nerve root. Here's one of them right there. The other one, who knows where it is. It's smashed flat back here, so horrible right leg pain. But that's a run-of-the-mill disc herniation. You can see it here. Anybody else see anything weird on this? Well, it's got a big ball right here, right? Oh, I do remember this guy now. Yeah, we did. This is called a Tarlov cyst. I'm not going to test you on that, but see how it's ate into the S2 vertebral body, because there's S1 and there's S2. So 99% of the time these are just incidental findings, but if they get big enough, they can start to cause kind of tailbone and sacral pain. This guy was asymptomatic. He didn't have any of that pain, but I was getting a little worried because it was irritating the bone a little bit. Another flaw, another committee flaw. Yep. So not only are the lamellae incomplete posteriorly, they're also thinner when compared to the lateral and anterior annulus. And that's yet another reason why we have these kind of posterolateral disc herniations. Remember the posterior longitudinal ligament, if I can draw that, remember that lives right about here, the strongest fibers of it. So that's why if we could draw the nucleus pulposus on this person, which we could see with a test called discography, but what he would probably look like is, you can't tell how big the annular tear is, but it would look like that, but it comes out lateral to the nucleus pulposus, or to the posterior longitudinal ligament. That pushes, that kind of prevents central herniations sometimes. Not all the times though. Those are the worst ones too. Well, the worst ones are far lateral, but it's not good to have a centrally located herniation. All right, the magic 65 degrees, very strange. So here, back to our cartoon here, these are the lamellae. You can see the black lines running in between these. See them right here? Those are actually the strands of type 1 collagen. And it's very interesting. We see this on other parts of the body. But if you draw a vertical line and then you measure the angle of kind of the direction that the collagen strands run, they're perfectly 65 degrees. And then what's even more amazing is that the next layer, the adjacent layers of lamellae, those layers run 65 degrees the opposite direction, or you could say negative 65 degrees. So the design is really, the committee scores points here because that design is great, because it gives great strength to the Annulus Fibrosus. But the only trouble is why it's hasn't evolved complete around the back is the problem with the human. Maybe because we haven't been walking upright long enough. Who knows what the story is. Magic 65. There's a close-up of it, and you can see how the adjacent strands of lamellae run different. The fibers of type 1 collagen run different directions. What about the histology of the Annulus Fibrosus? Instead of type 2 collagen, the cells of the Annulus Fibrosus make mainly type 1 collagen. So those black lines, those guys right there. I hope you can see my mouse, I think. Yeah, those are the type 1 collagen fibers. There's also some glycosaminoglycans between the black lines. So there would be GAGs in here. So there is a water content within the Annulus Fibrosus, but it's not super high. There are other types of collagen that we can't see or weren't drawn in there. But there's little type 2 collagen, type 5 and type 6 collagen also is seen in there. So the cartilaginous vertebral endplate kind of showed you what it looked like in that cartoon, but that is considered the third component of the disc. So the disc has 3 parts. It has a nucleus pulposus, annulus fibrosus, and a cartilaginous vertebral endplate. Because remember, there's a bony endplate. And you just have to look and see how the question's worded, because some people aren't that detailed. So if you ever see vertebral endplate, just assume that's part of the disc. And that is controversial. Even Bogduk says it's a close call because some people believe the cartilaginous endplate is part of the vertebral body, but most believe it's actually part of the disc. And there's again the cartilaginous vertebral endplate is right there. This cartilaginous vertebral endplate is made of, interestingly, 2 tissues. So it has a fibrocartilage which connects to the disc. So if I can draw that. So this part of the cartilaginous vertebral endplate is fibrocartilage. It connects to the disc. And then the hyaline cartilage connects to the bone. It's not a very strong attachment either. So if we go back and draw, let's see. So hyaline cartilage would be found here. It gives a little extra strength, but this isn't a very strong connection. This can be ripped off very easily until you get out in this region. These are called Sharpie's fibers. These lamellae here have special names. See how they're dug right into the bone? So that's the true ligament, the intervertebral body ligament. That is the ligamentous portion of the disc that connects the adjacent motion segments together. So those are a strong connection. Right, and there's Sharpie's fibers again, right there, which is just the outer lamellae, super strong. It is considered the intervertebral ligament, just like the anterior and posterior longitudinal ligaments. Those are all intervertebral ligaments. Ring apophysis is a secondary ossification center which caused the vertebral body to grow in height. So here it is right here. I think we've seen it in lab. Right, so it's a secondary growth center where the vertebral body grows in height, called the ring apophysis. Vertebral body top and bottom. So again, technically, you have to be careful with this word, vertebral endplate. Sometimes they're meaning the bony vertebral endplate. Sometimes they're referring to the cartilaginous vertebral endplate. So hopefully the board questions will be good enough to distinguish between those two. But there is a superior bony endplate and an inferior bony endplate. Those are the tops and bottoms of the vertebral bodies. All right, that's enough of vertebrae. Let's fall into one of my famous rabbit holes. We've been talking about GAGs or glycosaminoglycans, just kind of throwing that word around. We said the cells of the Nucleus Pulposus make those like crazy, very hydrophilic, grab water. The Annulus Fibrosus makes them. Those cells make them as well. But let's get into the weeds here on GAGs. So they're made by both Nucleus Pulposus cells, that should be, and Annulus Fibrosus cells. The main product of the Nucleus Pulposus cells are GAGs. It's not probably the main product of Annulus Fibrosus and cartilaginous vertebral endplate cells, because those are more cartilaginous main products, but yet it's made by all the cells of the, quote, disc. And the old AKA for GAGs, hopefully this won't be on boards anymore, but mucopolysaccharoid is an old AKA for them because they were first discovered in mucus, coughing up mucus. And mucous membranes have these GAGs. They're officially biochemically described as a heterogeneous polysaccharide chain. So what does that mean? So hetero means different. So it's different. Genus means kind of similar. So they're different, but they're kind of the same. Polysaccharide, that means sugars. So basically it's different sugars hooked together in a chain. And not too many different sugars, because they do tend to repeat in disaccharide units. So the molecules which resemble sugar or are sugar every 2 units is the same, and then they repeat. Oh, you're thinking disaccharides, I finally know a word. Some of you hopefully have had a little bit of chemistry and biochemistry. You know saccharides, right? You're thinking of these guys. Oh, disaccharides. Sucrose is made up of 2 sugar molecules. Galactose, not on here. Lactose, maltose. Those are the sugars that come to your mind. But these would do no good to have chains of these in the disc. Why? Because there's no polar groups. There's nothing to attract water. So to attract water, we have to have carboxylic acid groups, amine groups, or sulfate groups. And if you have those groups in your disaccharides, then you can grab water, and that's what the committee designed the disc to be highly filled with water. and so therefore we have to have polar groups. So those disaccharides won't work. Yep, everything I said. So it makes them, if you have a lot of these hydrophilic groups, or these polar groups within your structure, you will be like Proteoglycan Bob here and be able to grab tons and tons of water, which is just what we want. Also, the GAGs are negatively charged, so they do respect social distancing. They're going to be close to each other, but not that close because of negative and a negative repel, as we'll see here in a minute. So let's meet the GAGs. There's 6 types of GAGs that are in the body. They all have sulfur in them, so they're sulfonated, except for one oddball, this weird hyaluronic acid, aka hyaluronan. It's a strange GAG, but it's really an important one as well. There's only, with regard to the disc, we only are going to talk about 4 of them. And of these 4, they're mainly found in the Nucleus Pulposus. So let's meet the 4 GAGs. So you can, whatever mnemonic works. I like chick with the I not for my mnemonic. And the C stands for chondroitin. And you just have to remember 6 sulfate. And the H stands for hyaluronan or hyaluronic acid. The I doesn't mean anything. It just kind of separates these 2 C words. And here's another C for chondroitin 4 sulfate. And then we have a K for the keratin sulfate. So those are the GAGs that you'll find in the Nucleus Pulposus. A note, we've heard about chondroitin sulfate and keratin sulfate. Usually, mainly chondroitin sulfate, right? The vitamin industry has got that word drilled into our brains. And then they say, well, take glycosamine and chondroitin sulfate. Glycosamine is not a GAG, and they always screw that up on commercials. It's considered an amino monopolysaccharide, which is made from glucose. So you can convert glucose into glycosamine. And so that's just a little fun fact there. So GAGs of the Nucleus Pulposus, again, they contain superhydrophilic groups which attract water. Why are they so polar? Because the GAGs of the Nucleus Pulposus they all have mean groups, and those are hydrophilic, but they're not. They can attract a little water, but they're not that important. But the sulfate and the carboxylic acid groups, or the carboxyl groups, those guys are really, really hydrophilic, and they can grab water like crazy. And so that's what we need in the Nucleus Pulposus, because it's got to be 80% water, because under compression it acts as a ball bearing, and so we need it to be really, really full of water. Carboxylic acid or carboxyl group, remember it's a carbon double bonded to an oxygen and a OH group coming off it. Those of you who've had biochemistry know exactly what that is. And then a sulfate group is a SO4 group. Strongly hydrophilic, we said. Tract water like crazy. And this gives the disc in general its toothpaste kind of water slipperiness feeling. Who's the best trappers? Kind of said this. The chondroitin sulfate molecules have both carboxylic acid and sulfur groups on them, or carboxyl groups and sulfate groups on them. Keratin sulfate, it doesn't have a carboxyl group on it, like keratin sulfate or chondroitin sulfate does. So therefore, it can only bind half as much water. And the hyaluronic acid doesn't have a sulfur group on it, or sulfate group on it. So it binds about half as much water as chondroitin sulfate. Let's take a look for you visual people. So here's chondroitin 6 sulfate, and here's the molecule right here, this little disaccharide. And remember, all these GAGs are repeating, so this sugar, these 2 molecules, glucuronic acid and this N-acetyl glycosamine 6 sulfate, these repeat over and over and over again. But the key point is here. So they all have an amine group right here. But that tracks a little water. The key is right here. These chondroitin sulfate molecules, they have a carboxylic acid group here, and they have a sulfate group right there. And so these guys grab water like crazy. Chondroitin 4-sulfate is a little different conformation, but it's got the same carboxylic acid and sulfate group. So these are the water trappers, bottom line. Hyaluronic acid or hyaluronan, all it's got is a carboxylic acid group. There's no sulfate group here. It's still got the amine group. They all have amine groups. And the keratin sulfate has a sulfate group, but it doesn't have a carboxylic acid group. Alrighty. Okay, so GAGs, turns out most of them they don't like to be alone. They're social creatures. So there's 3 of them in particular that I call the sociable GAGs. They like to hang out and talk and drink beer maybe, who knows. But the sociable GAGs are the chondroitins, chondroitin 6-sulfate, chondroitin 4-sulfate, and keratin sulfate. So those guys always are near one another. And then, well, where do they hang out? Well, there has to be a place for them to hang out. So there's a GAG recreation center, not really, but there is kind of a place that they hang out called a core protein. And there's many different types. I'm not going to go into the weeds on these core proteins. They're actually used to categorize GAGs, but the core protein is the recreation center. But it's a stick, really, and these GAGs bind to this stick. The sociable GAGs do anyway. And yeah, so the core protein is also secreted in the nucleus by nucleus pulposus cells and annulus fibrosus cells. And cartilaginous and endplate cells all can secrete these core proteins. So yep, so the 3 sociable GAGs will join and mingle on a core protein. And this is an important point. Together they form what's called a proteoglycan. So proteoglycan is formed by the core protein and any mixture of the 3 sociable GAGs. Okay, that's an important concept. And of course, proteoglycans is what really makes up the nucleus pulposus. All right, so here's that bottle brush appearance. My voice is taking a turn for the worse here. But here's the 3 different GAGs, the glycosaminoglycans. So we have chondroitin sulfate all coming together, hooking up on this recreation center, this stick, which is the core protein. And then we have chondroitin 4-sulfate here and keratin sulfate here. So they're sociable, but they kind of like to talk to each other. And notice how they also keep the social distance. So they don't stick together. The author drew this one sticking together, but that would never happen. Because they're all negatively charged, they're pushed apart a little bit. But this is the classic bottle brush appearance. When you think proteoglycan, that's where that word bottle brush comes from. Where do you find proteoglycans beside the nucleus pulposus? In general, they're in the ground substance or extracellular matrix of different tissues, especially joints. So like all the major joints, your knees, your ankles, the synovial fluid are all filled with these GAGs to keep things slippery. And they're also found in the cell membrane, and I won't get into their function there. But yeah, if you don't know what a bottle brush is, there's a bottle brush, and that's how the first biochemists described these as, oh, look, they look like a bottle brush. And they really do. And the GAGs bound to the core, this is the core protein, this little double wire structure here. And these are the glycosaminoglycans, these bristles. And then the whole thing is called a proteoglycan. So got it? A proteoglycan is made of a core protein, mainly made of a core protein, and GAGs, glycosaminoglycans. Got it? Okay. is kind of everything I've said. There's some linker proteins, because we'll find out that some of these GAGs don't like to be alone. And we'll find out that they can hook up to a gigantic stick and form something called an aggregate. But we'll get to that. So there are some other proteins on here as well. So let's talk about now that we know what a proteoglycan is, there are different families of proteoglycans, and they have different functions, and they're found in different locations in the body. And the way the families are grouped is in accord with the gene that makes their core protein. The gene that makes their core protein through the central dogma, it's usually named after the core protein. And so it's usually the core protein names the family member of the GAG. For example, the Aggrecan family there's a bunch of different types of GAGs found, or all of these proteoglycans in the Aggrecan family are found in cartilage and disc. This is the one this is the family that we're considered, or that we're mainly going to talk about, because the types of GAGs and proteoglycans in the Aggrecan family are the ones that make up cartilage and disc. I should have probably put proteoglycan there, because these families are proteoglycan families. The Aggrecan family is made of proteoglycans. Proteoglycans are made from a core protein in GAGs. So put that on a note card. I know that's confusing. Put that on some note cards and get that in your noodles. It'll help you for this class and for biochemistry. So now we have a problem with this Aggrecan family name, though. So Bogduk uses Aggrecan family, so that's the way it'll probably show up on boards, at least for the spinal anatomy part of boards. But modern biochemists don't call it that. They call it the Lectican family. Lectican family, and I agree, it should be. Bogduk should change that, because it's confusing. Because one of the proteoglycans that is a member of the Aggrecan family, guess what its name is? Aggrecan. So that's confusing to have a proteoglycan named Aggrecan be a member of the Aggrecan family, because there's other Versican, there's other members of the family. So it's better to call it Lectican, so it's not so confusing. But that's always a confusing subject. So let's meet the Lectican family, or the Aggrecan family is how you're going to see it, of proteoglycans. And they each have a different arrangement of GAGs on their core protein, but their core proteins are different. And again, to be a member of the Aggrecan family, you have to, there's one rule, you have to contain chondroitin sulfate to be a member, somewhere on the core protein. The core protein is called Aggrecan as well, but you still can't be an Aggrecan unless you have chondroitin sulfate, either 4 or 6 or both. So let's look at some of these members. We're not going to go crazy on this, but again, here's the confusion. So the first member of the Aggrecan family is the proteoglycan named Aggrecan. This is the one made by chondrocytes in the cartilage of our body, in the disc. So all the joints have the proteoglycan named Aggrecan floating around and keeping things slippery. And again, it's confusing because Aggrecan is a member of the Aggrecan family. There's another one called Versican. This one's made by fibroblasts and smooth muscles. So this one's important for blood vessel wall function and in the dermis and a bunch of other tissues, but we're not going to worry about that. Biochemistry can teach you more about that. There's another one called Neurocan, and this one is made by neurons in the central nervous system, and that's important for function of neurons. Another one called Brevican. Remember, these are all proteoglycans, so they all are core proteins with GAGs attached to them. They're just in different conformations, and the core protein is a little different looking. Here's another CNS one called Brevican. This one's made by astrocytes. That's all I'll say about it. So Neurocan and Brevican are made in the central nervous system. I should have put more stars here because I do ask these questions. The one that's interesting here are the slurps, like a Slurpee. So slurp stands for small leucine-rich proteoglycan. This is worth talking about just a second for a slide here. So its core protein is, of course, we know proteins, or you will know, hopefully you know, but proteins are made from amino acids. Those are the building blocks of proteins. And its core protein is made up of these long stretches of leucine, the amino acid leucine. So that's where it gets the name from. There's actually two submembers of slurps. There's decarin and biglycan. And if you have a gene mutation in either one of these, and we'll just leave it at this, this is hot area in research right now because mutations in decarin or biglycan are found in people with type 2 diabetes, as well as people who have abnormal obesity. They don't eat much, but they just get obese. So they're kind of genetically programmed to be obese. So gene mutations for both of them. And there's one for tumor growth as well. So these are more clinically important. But type 2 diabetes is a huge problem. And it may have to do with a mutation in these in the slurp genes or its submembers. Okay, so again, the proteoglycan aggrecan is a member of the lectican family or the Aggrecan family. It's by far the most common type of proteoglycan in the intervertebral disc. It's the only one we really worry about. We'll take a look at the structure of this proteoglycan. It's made of keratin sulfate and chondroitin 4 and 6 sulfate. So those were the sociable GAGs, right? Those are the same sociable GAGs make up a proteoglycan. So its structure, it has a core protein. The core protein is not a straight stick though. It does have some straight regions. In fact, the straight regions are called E1 and E2, but it has some kind of curlicue regions as well. And those are called the globular domains G1, G2, G3. And this G means that's where the GAGs hook up and bind, and it also means that's where the water binds on these things. And so in general, it's considered a large to moderate sized proteoglycan. The core protein has about 70 GAGs attached to it. So let's take a look, kind of a nerd alert here. If you're a biochemistry fan, you'll probably like this stuff. But here's what it is. It's not a straight stick. The core protein has some straight regions. So this is the E2 domain. This is the E1 domain here. So pretty simple. And then we have these curly kind of squiggly regions. So we have G1 domain, G2 domain, and the G3 domain. The G1 domain is what binds. Some of these can bind to hyaluronan to form this gigantic aggregate molecule. So that's where the binding occurs. There's a linker protein I took off there. But the point of the slide is to show you that all the GAGs actually bind to the straight E2 domain and to the E1 domain. And if you want to get specific, the E1 domain, which is this piece right here, it only binds keratin sulfate, whereas the E2 domain can bind both keratin sulfate and both of the chondroitin sulfates as well. All right. So wherever these GAGs are, this is where the water binds. So that's exactly where the water binds. Bottom line, water binds on the straight domain E1 and E2. The curlicue domains we're not going to worry about. All right, and there's what in a real one would be kind of twisted like this because they're really long, right? And they have you can see the water bound to all these GAGs here. And that slipperiness and the repelling, these negative charges are repelling, so that's what gives it that kind of slippery feel too. All right, so where do you find these GAGs anyway, or these proteoglycans? Where do they live? Kind of said this already. In mucus, in the nucleus pulposus, synovial fluid, in the vitreous humor of the eye. So the eye has some jelly in it made of proteoglycans. Also, when you get to gross 2, you can, when you get to the cadavers, you can feel in the pericardial lining. If you pull the pericardium off the heart, you can feel how slippery it is. You're feeling gags when you feel that. Same with the pleural cavity and around the lungs. You can feel that slipperiness still in the cadavers, and that's because there's gags in there. Okay, so now to go one step further, some proteoglycans are also sociable. We said GAGs are sociable. They like to hook up on these sticks of protein called core proteins to form proteoglycans, but some of the proteoglycans are also sociable. So they can bind to another stick, and this is where the oddball glycosaminoglycan called hyaluronic acid, aka hyaluronan, that's where its job is. So it's a social gathering place, not for GAGs, but for proteoglycans. So proteoglycans can hook up on a stick of hyaluronic acid and make a gigantic, humongous molecule, which is called, confusingly, proteoglycan aggregate. So don't confuse proteoglycan aggregate with a proteoglycan aggrecan. And don't confuse that with the family of Aggrecan. See how confusing it is? That's why it's better if they don't, they get rid of that family name. But anyway, so aggregate, look for that gate. That means it's a gigantic molecule. Now these aren't in the disc that much, but these are in your knees, your ankles. They're super slippery molecules. And here's a cartoon of one. So here's the little bottle brushes we talked about. All right, so those are proteoglycan aggrecan molecules here. There's the green core protein. So they're sociable. Some types of them are, and they bind to this stick of hyaluronic acid. And yeah, so these are called proteoglycan monomers. Actually, the aggregate is not sociable, so we don't see that in the disc. But other types, like Versican and the other CNS one, they're more bindable like this. But anyway, bottom line, aggregate molecule is this gigantic creature right here. Okay, it can even contain several species of proteoglycan, and yep, it's said to have a hyaluronic acid backbone. Yep, we could say that. All right. Now again, the aggregans aren't very sociable. They don't typically bind to hyaluronan to form this giant aggregate molecule in the disc. So they prefer to be alone. So you won't find these aggregans in the disc. You may be a couple of them, but not many. So yeah, they're more commonly found in articular cartilage. Right here is electron microscopic view of one here. And yeah, that's an aggregate. Okay, enough of that, biochemistry. But let's get into some biology. So enzymes of the disc. You might not know anything about these, but they're really important, especially in people who have degenerative disc disease. So there are enzymes that are made by the cells of the disc. They also make proteoglycans, of course, is probably their main product, but they also make, they make those core proteins, they make hyaluronan, but they also make enzymes. And that's true for articular cartilage as well, your knees, ankles, elbows, and stuff. So there are enzymes. They are important for kind of fixing tissue that gets broken. If you had a heavy squat day, you're squatting 550 for threes or something, you've damaged some of the tissue inside your disc. Who's going to clean that mess up? Well, these enzymes, and they're called MMPs. So they're called matrix metalloproteinases, lots of stars. You have to know these because this is the cause of one cause of degenerative disc disease. A non-traumatic cause of degenerative disc disease is when we get mutations in things that control these, as we'll see. So these are the key enzymes that maintain or these are the key maintenance, I guess you call them maintenance enzymes of the disc, are the matrix metalloproteinases. There's 3 groups of them. So MMP1 is called collagenase, and you need to know these. MMP2 is called gelatinase. MMP3 is called stromolysin. And why it doesn't have an ace on it, I don't know, because it's a sinner, because it can be evil. And this is the cause of one of the proposed causes of pathological degenerative disc disease. Just someone who has never had any trauma or nothing wrong with them, but they have horrible looking discs and have all kinds of pain, and it's usually in the family. So stromolysin is a very wicked dude. So let's meet them. Collagenase has really one function. It will chop type 2 collagen up, but it won't make them small digestible particles. It just chops the main type 2 collagen up that's been damaged and is floating around. We need to get rid of it. So it'll chop it up into large fragments. Then gelatinase will come in and chop those large fragments into small fragments, and usually they're harmless when they're small fragments. But then we have stromolysin. So this is and strom, like poem, strom, stromolysin is the most destructive of these guys. It's used for cleaning up all the debris. So stromolysin will take these small fragments, clean them up. It'll take on big fragments that weren't cleaned up. It'll take on anything. But it loves proteoglycans, and that's the one it usually goes after quite aggressively, to the point where the proteoglycan and even the Aggrecan or aggregate molecule is destroyed by these things. And it doesn't stop there. It can cut up type 2, type 9, and type 6 collagen. It can chop up fibronectin. It can basically destroy your disc. Just about anything in your disc can be destroyed by stromolysin. It is a sinner because it is evil. It's actually, we need it though, but the trouble is when it gets out of control. So MMPs normally remove the damaged components of a disc after you just squatted 550 for 3 or whatever. But so because the disc normally isn't that destroyed all the time, there are usually not many of these MMPs floating around in the disc. So that's good, because you don't want them, if there's too many of them, what's to stop them from going after good tissue, going after the lamellae? And that's the problem. That can be the problem. But normally you don't have a lot of these things. How come you don't have a lot of them? And they are dangerous, so we need ways to keep them under control. So there's 2 sets of molecules, 2 sets of biomolecules that keep them under control. So we have MMP activators. So normally MMPs are secreted in inactive forms. I'm not going to make you know those names, but they just aren't secreted by a cell and they start ripping things up. They have to be turned on. It's a regulatory system. So we have MMP activator, and that one's called plasmin. So that's really important, plasmin. So that'll turn on, that'll check, okay, do we really need these MMPs turned on? Do we want to let loose these little tornadoes? And it looks around and says, oh yeah, we got a lot of damaged tissue here. We better turn them on. So that's its job. It turns them on. But then we have the TIMPs. TIMP1, TIMP2, TIMP3. These are the MMP inhibitors that are also floating around. And this is where problems can occur. So when the job is done, when they've cleaned up the damage, maybe you've ripped the inner 5 lamellae and there's fragments of type 1 collagen and type 2 collagen and gunk floating around, needs to be cleaned up. Great, plasmin will activate the MMPs and they'll start cleaning up. But now it's time to turn them off, and so the TIMPs will survey the land and say, okay, it's time to shut you guys off. And normally, no problem. The TIMPs will turn off all the MMPs. But And this is what we think pathological disc degeneration is. There are known gene mutations which can occur in TIMP 1, 2, or 3. And people with pathological or premature degenerative disc disease have a high level of TIMP gene mutations. All right, and if you have a mutation in the TIMP gene, well, you can't turn them off, right? Because plasmin turned them on, but who's going to turn them off if the TIMP gene is damaged? And that's the problem. Sometimes they just run wild, and they destroy not only after the bad tissue is destroyed, the good tissue is now destroyed, and you get degenerative disc disease. All right, and then sometimes you have genes that overexpress the MMPs. You have overactive genes. So that's another problem, but it's not as prevalent as these TIMP gene mutations. Stromatolysine is, if that one gets loose, you're going to have degenerative disc disease. All right, so that's all I'll say about that section, and just a little wiring. I think we know this, but let's really get a little more into the weeds on the wiring of a motion segment. So we know that sinuvertebral nerves are really important. We know the AK already is the recurrent meningeal nerve. Another AK is the recurrent nerve of Leucina. So they're sensory and nociceptive and proprioceptive. They do carry sympathetic fiber as well, as we'll see. Then the medial branch of the dorsal ramus. That's the one that supplies the facet joint or the Z joint and the multifidi muscle. So let's get into the sinuvertebral nerve again, recurrent meningeal nerve, strong AKA. Most authors use sinuvertebral nerve. Kramer goes against Bogduk and uses recurrent meningeal nerve, so you have to know both of those. And it is considered a branch of the ventral ramus. I've asked that question before. Somehow that I've tried to see if you knew that the recurrent meningeal nerve was a branch of the ventral ramus. It's not a branch of the spinal nerve. It's not a branch of the posterior ramus. It's a branch of the ventral ramus. Ventral ramus is what gives rise to the sciatic nerve in the lumbar spine and the brachial plexus in the cervical spine. So its main job is nociception. It has the ability to carry the sensation of pain. It also, because it has sympathetic fiber, it can control the diameter of the blood vessels in the outer portion of the nucleus pulposus. And there's a weird proprioceptive. It may help with proprioception, which are nerves that tell your brain where your body is in space. Okay, so what does it innervate? So, there's the little weird monkey. I forget, is it a screech monkey? There's like a little weird. He's surprised. So this is where the confusion. When somebody says, they come in, Doc, I have this all the time, my patients. It's L5. I can feel my pains coming from L5. Or, Doc, I have L4 pain today. Or even chiropractors say, yeah, your pain's coming from L4. You don't know where your pain is coming from because two segments are wired together. And even the sacrum, we've talked about this in the SI joint. L4, L5, SI joints are all kind of wired together, so you can't tell really where the pain is coming from. But officially, so the posterior and posterolateral annulus fibrosus is supplied by sinuvertebral nerves. But here's what you may not know. Posterior and posterolateral annulus of the level above is also supplied by the same level, sinuvertebral nerve. We'll see a picture in a second. Then the anterior portion of the thecal sac is supplied by the sinuvertebral nerve, and the posterior longitudinal ligament. The PLL is also supplied by the sinuvertebral nerve. Guaranteed one of these questions is coming. So this isn't the best drawing in the world because they made a really skinny little ventral ramus here, but it's kind of get this out of the way so we can see the main players. There's the sympathetic chain that you know. There's sympathetic ganglia. There's sympathetic fibers run within this gray ramus communicans down here in the lower lumbar spine. And then here is the sinuvertebral nerve. So it comes off the ventral ramus. It also has a connection to the gray ramus communicans. That's where it gets its sympathetic fiber from. But here's what you don't know. So right under the pedicle, it splits into one branch. Sometimes it's called the descending meningeal branch. That supplies the L4 disc. But it also has another branch that goes up to supply the L3 disc. So sinovertebral nerve, if you have a tear in the L3 disc, some of that pain may come in through the L4 nerve root here, the sensory root, and that's why you can't tell these apart. So each disc is at least double innervated. Got it? And there's also a little crazy plexus we'll look at next time here. So, yep, that's the story there. Right? And we've seen this picture in lab, but I don't know how strongly I tested you on it, but now we can see a little bit more of that same setup here. Right? These little lollipops have the ability to transmit pain. The little lollipops are also plugged into this pink PLL, posterior longitudinal ligament, and the little lollipops are plugged into the dura of the dural sac, or aka thecal sac. We got it? But what we don't see is the branch that's coming out of the plane of the page and supplying L4. We just see this level, so it's more complicated than meets the eye. And I don't need to go through this again, I don't think, but there's a sensory and a motor root, and they're not always in this order too. They can be in any order they want. Maybe sensory's back here, maybe motor's up front. There's the spinal nerves where they come together and splits into a dorsal ramus, ventral ramus here. Medial branch is very important. That gives life to the Z joint. That should be review. Everything I just said, and that's enough for your brains today.